Welcome back to the Impact Storytelling Podcast. I'm your host, Davina Probst-Saavedra, Ocean Conservation Storyteller and Organization Director. Today, I'm joined by Julia Giebichenstein, a PhD candidate at the University of Oslo and a member of the Nansen Legacy Project. Her research focuses on the seasonal and interannual variations of legacy and emerging pollutants, including persistent organic pollutants and mercury, across the northern Barents Sea pelagic food web. She combines pollutant measurements with dietary tracers, such as stable isotopes, to link pollutant levels with diet and track how pollutants move from small plankton to top predators like polar bears. Welcome to the podcast, Julia. It's great to have you here and talk with you about your research. How did you first become interested in marine science and ecotoxicology, and what inspired you to pursue research on pollution and climate change in the Arctic?
Hi, Isabina. Thanks so much for having me. Very exciting. So to be honest, I started quite I feel like how many young kids start when they want to become marine biologists from our generation. I watched Free Willy, and I really, really wanted to be an orca researcher when I grow up. But I grew up in Germany, very far away from the ocean, so it was a bit like, okay, how to do this? So I looked into, universities where I could study marine biology, and there are just a handful in Germany, less than a handful, I think. And it's quite competitive to get in, and I didn't have the best grades ever to do this. Also, being just coming from school, money was an issue. So I think all of this led me to just, okay, I'll stay close to home. So I started studying in Düsseldorf. It was a biology bachelor's degree. But luckily enough, there was this professor who offered marine biology classes. So I was like, oh, this is going to be the door into marine science, hopefully. So I wanted to try that and also see, because it has always been a dream of mine. I've been passionate about the ocean. But up until then, I had never really any experience, like hands-on experience of ocean-related research. So it was also to trying to figure out what fits me and if it's actually my passion. So I took the courses which were offered by Professor Christopher Bridges, and he offered field trips to northern Germany to visit the coast there, as well as Scotland and South Africa and Namibia, and all of this was so exciting, and just showed me, okay, I'm on the right path. Like, this is really what I want to do. And then after spending some time in that lab, kind of, and also joining these field courses and in his team, I met one of his PhD students, and that was just being in the right place at the right time kind of situation, because he was like, oh, I need help for my PhD work up in Tromsø, in northern Norway. Would you be interested in joining? And I said, sure. I have never heard of this place before, but it sounds, interesting. So I joined him, and that was the first time I went to northern Norway. And since then, I don't know, everything changed kind of. So that was a very pivotal moment, because before I hadn't even thought of ecotoxicology. Like, that word even was like, I don't know what it means, but kind of coming to Tromsø for that research was where things started to change, I think, and where I became interested in what I'm doing right now. And so at that time, it was also because I researched where I could kind of be close to orcas, and I knew in Tromsø is where they are in the winter time. So while I was there during 2015, 16, 17, the fjords just around Tromsø, they were bubbling with whales. So it was humpbacks, orcas, and I started working also as a researcher there, and also guiding. So this was like the first step to marine science and ecotoxicology.
Sounds amazing, even your first experiences and all the field trips before. Norway and then in Tromsø, and beautiful that he got to see all the rails.
Yeah.
So I guess this is what motivated your transition from studying biology in Germany to conducting field-based Arctic research in Norway.
Yeah, exactly. And it has been it was never this moment where I woke up and saying, oh, I'm going to move to Northern Norway to pursue my dream. It was more that I was introduced into this world by the PhD student that I work with, Gwendolyn Göttler is his name. And that was just kind of a smooth entryway from there. And then I realized Tromsø is just such a magical place in a way, because you're surrounded by, I mean, you've recently been there, so you're just surrounded by this incredible nature, and you have the northern lights, you have the whales and the fjords, and on top of that, the community up there is just fantastic. So I think it's quite international, like, especially when you come in as an international student. So the connections that were made, it made life very easy up there, although the winters are a bit rough and long and dark. But that kind of made up for all of this. Then, after finishing my bachelor's, I really wanted to go back to Tromsø. And I saw that there was an Erasmus exchange program from the University of Düsseldorf, which I then did during my master's degree. So that was also the first time while I was in my master's, I took an Arctic marine pollution class. And I had never heard of chemical pollution, or not never heard, but I wasn't very aware of it, of chemical pollution, that was a thing, especially not in the Arctic. So this was a really eye-opening course. I remember one of the teachers telling us, oh, have you ever used this microwave popcorn? You get the popcorn and you get the corn in the bag, put the bag in the microwave, you eat it, and but in that lining of that bag, there are these forever chemicals which can have detrimental effects on our health. And she was like, this is stuff that we use, that all of you use. And she just made these really hands-on examples of how chemical pollution is kind of all around us. So that blew my mind. And I think this was a second pivotal moment for me where I realized I love orcas and I always wanted to be this kind of orca researcher. But at the same time, while I was guiding, we also had so many tourists coming to this remote region. And I just saw the impact of the tourism, what that had on the local communities, also the impact on the nature, because all of a sudden you have those spots that are just not visited by people very often, which are swamped by people. And I mean, people have been, I don't mean that anybody left any trash behind or so, but it was more like that activity must do something with that local environment. So I started questioning also my role in this, and I kind of wanted to find out, okay, what is our impact on the oceans? And with the ongoing climate change, how is that all interacting? So yeah, I guess that's how I moved away from Germany and into ecotoxicology and the Arctic.
It's super interesting. I think there's really a lot of things that we're using and we don't know how harmful it actually is. And also when I was talking with Claire, I said it's crazy that there are not more regulations and that they're just changing one little thing and then it's okay to use again. It's it's really everywhere. Also in the Arctic, I think in every place of the earth you probably can find something that shouldn't be there.
Yeah, exactly.
Could you describe the methodological challenges of fieldwork in extreme Arctic conditions, as you have been on many field expeditions in the Arctic?
Yeah, I would love to. No, because it's so being in Tromsø is this very unique research environment that's people are just very approachable, so it's easy to connect to people. So for my master's thesis, Initially, I wanted to work on plastic pollution because that was what I always saw. We did lots of beach cleanups. We saw at that time there was this whale that's stranded in Norway that had its stomach full of plastic. So it was everywhere in the media. So kind of plastic pollution is what people are, I think, a bit more aware of, and I was more aware of. And so I tried to get into that and reached out to lots of people. And then one person directed me to a different person and said, they're starting work on this Arctic fish, and they're including climate change and oil pollution, which is also a very important topic when it comes to pollution research. So I got in contact with Yasmin Nagang. She's a tox professor at the university in Tromsø, and her PhD, Morgan Bender. We would then work together on that project. And to start the project, we needed fish that we had to catch in Svalbard. So we needed to board a big ship, catch the fish, and bring them down to Tromsø. And this was so Yasmin had asked me at the time, do you want to join the cruise? But we're leaving in 10 days. So to me, it was a no-brainer. I didn't have children at the time, so it was very easy to say, yes, I can come. So that was my first experience joining a research cruise, and I was just baffled. The amount of work that goes into planning the research cruise, the logistics to get everything on board, all while it's minus 10 degree and snowy and just not very comfortable to be outside. And then once you are on board, you just have to be sure that everything you need has been packed. So it's just this very unique environment. So for my first expedition, for instance, we went to Svalbard to catch the fish, and that was during the polar night. And polar night means there's 24/7 of darkness. There's no sunrise, no nothing. We're kind of going to this black void when we're on the ship, and being there 24/7, having to know, okay, I have to just function, everything has to work, but then things will happen that nobody has accounted for, and we won't always work as we have plans. So we have these daily meetings to update each other and to adjust the plan as we go. And we hit a storm at some point during my first expedition, and I get seasick. I didn't know how violently seasick I could get, but I experienced it then. But it was also comforting to see I wasn't the only one, because apparently we hit one of the worst storms that expedition, which usually goes once a year at that exact time of the year. So people have had quite experience, and I was one of the newbies on board. But seeing apparently it was quite a bad storm, because it was at least half the people who got really sick. And even one of the crew at some point had to take a break. And that was reassuring, because I thought, oh, I'm on my first expedition and I'm really, really sick and I can't work. And then it also feels like, because it's so precious, this time on board, it's so much money and resources and fuel and everybody's time that goes into this. So you also feel like, okay, you just have to do your job, and you have to do your job well. And if you can't do it, that feeling was just like, oh no. It was heavy, but at the time, we just couldn't do anything. So we waited out a storm in a fjord. But then at some point we had to leave because time was running out and we had to get home. Once we got out of the fjord, and we had just done a trawl and started to process fish. So we had all these fish lying on the tables, trying to sample them. And that we went out the fjord with the ship, and the storm hit and everything just went sideways. You couldn't walk. You had to really hold fast. So those samples, for instance, they were difficult to keep. So we had to find alternatives for this. And this is one example of how Arctic research can be super challenging. And I had incidents where I was trying to, had this metal pump that kept freezing. And I had to take off my gloves, adjust stuff. The crew had to help me, and they took off their gloves. So we had to be so careful not to get any frostbites. And I'm pretty sure at some point someone got frostbites, because it was just insanely cold with the wind. It was, minus 30 or so with wind chills we had sometimes. And it's just so uncomfortable. So while you do this, you always have to keep in mind, okay, this is soon over and it will work somehow. And what is amazing on these ships is that usually the crew and the other scientists, everybody's so supportive of each other. So everybody kind of wants you to succeed, and we want each other to succeed. And this is insanely helpful. So people just come outside to help you, and you get your stuff done, basically. And so the technical difficulties are just, they're omnipresent. And while you do this research, you just have to keep in mind, okay, I'll adjust as I go. And I might not get all the samples that I wanted to. Maybe I get something else, which usually leads to other exciting questions. We had incidents. We wanted to catch amphipods, this is the food that the polar cod eat. Polar cod are super important fish in the Arctic ecosystem. I studied them during my master's thesis, and now they're also part of my PhD thesis. And so we tried to get the food of this fish. And we had these little traps. They're plastic pipes, basically, that we wanted to attach to the sea ice. So we went out, super proud, my colleague Robin and I, and deployed those traps. And then we were back on ship. And at some point, I think it was just a couple of hours later, if at all, one came to us and they're like, "So there's a polar bear munching on your traps." And apparently, we put bait in those traps, and maybe it had attracted a polar bear, but we don't know for sure because the bears anyways come to the ship and kind of explore it a bit. They're very curious. They don't have any other predators, so they're not fearful or anything. So they come very close to the ship, which also means we have to have polar bear guards on board at all times when we do sea ice work.
Wait, can they come on board?
No. But we were scared. I think we had one incident where a female polar bear, she came really, really close. But the ship is too high, and I don't think they are that interested in us either. And also, polar bears are protected, so we are not allowed to do anything to them unless they are starting to harm us. So we just stood there and waited while the polar bear came. I could have, if I wanted to and really stretched out, I could have probably touched her. So she was one of we've seen many bears around, but she came really, really close. And bears eating our equipment or trying to eat it is also, we had 2 incidents where that happened. One with our traps, and I think it was the same bear then who went, and we had some device on the ice that measured that had traps in the water attached, basically. And it was attached with buoys, and the bear started to eat the buoys. And then there was another thing that he tried to eat, which was also deployed on the ice. So at some point we had to call the governor in Svalbard to check if we can fire a warning shot to scare the bear away from the equipment, because we have to protect the bear, because if he ingests anything, could choke. And also the equipment, which is also quite costly to deploy and have there. So it's a safety issue for everyone involved, I guess. So we tried to scare the bear, and it was so fascinating to see because we got the go from the governor at some point. So we fired this warning shot into the air. It's very loud, very powerful noise, and the bear, he ran for a couple of meters, like 50 meters maybe, and then stopped. That just shows they are the apex predators in this environment. They're not really scared of anything.
Yeah, they probably don't know what the shot means because they're not used to getting hunted or anything. So it's just a loud noise for them.
Exactly.
And if they don't see danger, they just say.
I don't know if they know what danger is at this point. Like, through their life, probably they're just scared of the other bears. But yeah, so that was incredibly fascinating to see. And so the methodological challenges involve numerous ways of how things just could go wrong. I I lost a box at some point because when we prepared for the cruises, I had to ship a box or ship multiple boxes from Oslo. Nine arrived and the 10th was missing. So then I was stuck on a boat in Svalbard already trying to leave for the research cruise, but I needed what was in the box to do my research. Um, um, and then I very desperately called my supervisor, Katrine Borgo, and I said, the box contains these filters that I need for my water pump. What am I supposed to do now? And it was my first cruise for my PhD. And I was just desperate. And then she said, yeah, okay, don't worry. We got this. This will be fine. So you just tried to do something else. But then on board, we at some point needed to have other parts for the boat delivered by helicopter. But by that point, we had left. We were in the Svalbard water somewhere. And then a helicopter anyways came to that boat, and my supervisor heard of it. And our cruise leader, Marit Rijkstad, she was an amazing cruise leader and also the project leader of the project that I was part of. She coordinated with my supervisor here in Oslo to have those filters that I needed loaded, shipped from Oslo, loaded on the helicopter, and then brought to this remote island somewhere in the high Arctic so that we could pick it up and I could sample. And it was just such a brilliant way of showing, okay, you have to be super adaptive. Everything just has to, you just have to be aware, like use the opportunities that are presented to you in a way. So it was really cool to see, and everybody worked so well together, and it was just very quick and cool. So this was a very happy ending to a chaotic start, I would say. So things will always work out somehow, I think. But there are so many challenges that people probably will encounter, or I encountered.
I think always working in nature, there are challenges, and then, being human, having to travel. So
Oh, I had this one friend, because those cruises, they went on for 3 weeks, and she lost her luggage in transit. So while she was traveling to Svalbard, her suitcase got lost. So that was also another thing. Like then you're in the Arctic without your stuff. You don't have any clothes. You need lots of clothes, of course. So that was also a hassle for her at that point. So things, life just happens, and you just have to be prepared and have one of the technicians on board, Annette Wold. She always had this MacGyver kit with her, where she had all sorts of tools, things to sew, because if a net breaks, for instance, you have to sew it together, or replacement nets. She had everything in that little bag, or not the little bag, it was a big bag, but it's quite fascinating.
Great. I also have like the bag like this for when I go filming, with all kinds of stuff inside that I could need if something breaks down.
Yeah, that's just so useful and smart to have. Good.
Can you tell us about the overarching scientific question that guides your PhD research now?
So after I moved away from Tromsø, I was initially supposed to do a PhD there, but then this PhD project came up at the same time, and it was just the absolute dream, where I was able to be part of a large project. This project is called the Nansen Legacy Project. It's finished by now, but we are still a few PhDs who are finishing now. And within that project, the overall goal was to study the Barents Sea. And the Barents Sea is located north of Norway and northwestern Russia, and like on the east of Svalbard. So this was the main study area of that project, and it was quite a big project. I think we were around, I forgot the number now, but I think it was around 80 or so early career scientists, so PhDs and postdocs, plus all the senior scientists and researchers. So it was a really, really big project, and across entire Norway. So we had different work packages, and within my work package, we focused on human impacts in the Barents Sea. And then in my PhD research specifically, I want to understand how the Arctic seasonalities, so this drastic contrast between daylight and the complete absence of daylight, and with that, the changing sea ice condition, changing temperatures, and changing, especially the changing food abundance, um, how all that shapes the contaminant distribution in the food chain. So it's it spans like my food chain kind of spans really from tiny critters like copepods to polar bears. So it's a quite broad approach what we're doing here. So and one of the yeah, and so we're trying to understand, so are there specific times in the year, for instance, where the pollutant concentration is higher in the food chain, and how is that impacted then by the body condition of the animal, all of those questions. And I tried to answer this by going on 4 different research cruises across 1 year. So was the plan. And have spring covered, summer covered, autumn and winter. That's partly worked because it was interrupted by COVID. So we had to adjust a bit for this. So we had 1 year break, 2 research cruises, 1 year break, and then 2 more, where I was able to then collect data on, those copepods that I mentioned, which are like tiny crustaceans. They're everywhere in every drop of water we find. And then in any body of water. And also the fish we had these little krill. Krill are, I think they're kind of a hot topic now. At least I've been hearing more and more about krill from Antarctica and what big of a resource they are there. So it was also part of what I looked at. And then we had fish, and then I received samples from other people from polar bears and seals.
Mm. And what makes the Barents Sea such a valuable natural laboratory for studying human impacts on marine ecosystems?
Yeah, that's a super interesting question. So the Barents Sea, I mean, it's also because I study in Norway, so I'm based at the University of Oslo. So, and the project that I'm involved in is its main focus area is the Barents Sea. But so the Barents Sea is quite unique because it's kind of this entryway into the high Arctic. It's a shallow shelf sea. It's quite accessible, and it's where it contains a marginal ice zone. So here, open ocean and sea ice meet, which varies across the year depending on the season. So you'll have more sea ice in the winter time, less sea ice in the summer, and because it's influenced by these southern currents from the Atlantic, which are warmer, and it exports cold water from the Arctic. So it creates this kind of unique ecosystem with lots of biodiversity and lots of production. So compared to other Arctic areas, this is a highly productive sea, which then provides lots of food for animals to live and thrive in there. And I think it has one of the densest seabird colonies on the islands in the Barents Sea. And it's just, a very productive, unique region, but also has high commercial interest. So the fisheries are quite prosperous, and they're also very well managed in the Barents Sea. We have an interest in oil. So there are oil reserves that could be used in the future. And then, it's those two mainly. And I guess also deep sea mining will be in our question there in the future. So, but also having in mind that the Arctic warms almost 4 times faster than any other region on this planet. We experience these drastic changes there. So we have lots of sea ice reduction and all of these things. And the increase in temperature, of course, will lead to more ship traffic in the Arctic. We already see it, and it will probably just get more and more, which is not just a commercial ship, um, either for fisheries or tourism or transportation. So all of these things will happen in the future Barents Sea. So what we also why we also chose the Barents Sea then is to have uh, good baseline data to offer to inform management and say, okay, this is the current status of the Barents Sea. There are also parts in the project who modeled the future Barents Sea and what is it going to look like. So all of these things, I think, make the Barents Sea super interesting, also because of this high commercial interest amid this ongoing climate change.
And why are persistent organic pollutants, mercury, and contaminants of emerging Arctic concern important to monitor in the high north?
That is something I wasn't even aware of until I started my studies. So when I first came to Tromsø and took this pollution class that I just mentioned, we had these very hands-on examples of how chemical pollution is kind of all around us in the food packaging we have, in the electronics that we use, in the care products. The list goes on. Children's toys, don't even get me started, on the playgrounds and the tires. It's an endless list. But in the Arctic especially, the Arctic acts kind of as a sink of global pollutants. When you think about it, the Arctic, it seems quite far away. It's on top of our Earth, but it seems so far away from most of us, especially for me. I come from Germany. I had close to zero thoughts of the Arctic before I started moving, like living there, studying there, and working there. And chemical pollution, whatever we find up there, has to be quite persistent because it hasn't broken down. It is transported from lower latitudes because there's no large industry in the Arctic. So whatever we find there is from more southern latitudes. And I'm not excluding local sources. We know by now that local sources, like local settlements in the Arctic, also play a role in the local pollution. But most of what we measure up there comes from more southern and more industrialized regions. So what the Arctic also does is it kind of acts as this canary in the coal mine for global pollution, because whatever we find there should be some sort of warning sign and red flag, because it seems to be quite persistent, hasn't broken down on its way. And Arctic monitoring, I think it started when the indigenous communities had these sky-high levels of persistent organic pollutants and mercury, which can have adverse health effects. Because many indigenous communities still rely on hunting whales and seals, and these animals are all quite high up in the food chain. So that means, over their lifetime, they had a lot of time, kind of, because they're long-lived species. Over time, they had lots of time for contaminants to accumulate in their body and just build up. But also, these animals, since they sit kind of at the higher level in the food chain or at the top of the food chain, they have been eating all the things that have probably already also ingested contaminants over their lifetime. So they usually have the highest levels that we see. And if these are then being eaten by the humans will have even higher levels. So because while energy that we get from our food is being burned, the pollutants, they are retained in the body, many of them at least. So this has been an issue that we saw the northern communities having these really high levels, and the question was why. So then monitoring started, and it's super well established now. So now we know where to look and what to observe, kind of. But it's always an arms race with the industry.
And how do seasonal lipid cycles in Arctic organisms influence the accumulation and release of pollutants on the food web?
Yeah, so that's a great question because this is kind of also the core of my research and what I'm interested in figuring out. So when I go to the Arctic, I need multiple layers of clothes to be warm, not get any frostbites. Um, and Arctic animals, they don't do that. They can't and don't have to do that because they have these large lipid stores in them. And it's so cool to see. There are some animals that like it's a copepod, tiny, where you could really see when you put it under the microscope, you see these oil droplets in the body, and the oil sac it has. And it's when we caught them, there were oil droplets on the water surface because they're just so fat. And when they die, of course, the lipid gets out. So they're insanely fatty species, and in the Arctic, and also the whales, they have their blubber to insulate them. Bears, they also have this adipose tissue. They have the thick fur, with the hollow hairs to keep them warm. So there are so many cool adaptation when it comes to Arctic animals. But what most of them share is this fat, is the high lipid content, and many animals, not all, they have adapted a seasonal feeding strategy. So what happens in the Arctic spring is that the sun returns finally from from its 6-month long break and rises above the horizon. And that triggers this cascading effect of an algae bloom first. So you have phytoplankton growing in the water, and then the zooplankton emerge. So those tiny little critters like the copepods that I mentioned, the krill, all of these come up to feed on the algae. And that again attracts more species. So what all of these animals do at that time of the year is they really need to build up their lipid stores for the next winter and to be able to reproduce. So what we see then during the spring and summer months is that the lipid really increases in the animals, especially towards then the winter. They're stocked, I would say. So they're just really fatty, really lipid rich, high energy, containing meals for each other, I would say. So then what happens during the winter, the lipids are being used up by the body because there's less food available, the sun has vanished, so there's less phytoplankton in the water. So by now, we also know that this polar night period, it was long thought to be dormant and it's like non-vital almost. But we know nowadays that's really not the case. There's so much life happening at this time of the year. And I joined a couple of polar night cruises, and it was just amazing to see. There's so much life at that time of the year and so many baby copepods. So it's just there's lots of stuff happening during the polar night. Um, and then when we look at those lipids who vary this much throughout a year. So during this winter time, while the animals reproduce, they use up their lipid stores. We were wondering what happens to those pollutants because most of the persistent organic pollutants, they are fat loving. So that means they kind of attach to the fatty tissue. And that also makes Arctic animals more vulnerable to this kind of pollutants than, for instance, animals that live in Germany, because just of their higher fat content. So they can accumulate lots more. What happens at the time when a polar bear, for instance, starves, they have to remobilize they use the fat, and while they do that, the pollutants in that fat is being remobilized and released into the bloodstream. And there it can move to the livers, to the kidneys, and can cause toxic effects. So at that time of the year, we also then see or assume they will be a bit more vulnerable to this kind of pollution. And we've been wondering then, okay, how does it work for the entire food web? Do we see those fluctuations in pollutants, which follow the kind of the energy, the fat content? Do the pollutants fluctuate at the same time, or is there a delay, or all of these things? So I think this is why it's super interesting to consider lipid dynamics in an organism, the body condition, and how pollutants then kind of follow that pattern or not. And this is what we tried to find out with our research.
In your research, you use tools like stable isotopes. How do these kinds of dietary clues help you connect contaminant levels in an organism to its feeding habits within the Arctic food web?
Yeah, so stable isotopes are really cool and useful. So what they do briefly is, in my research, we used bulk stable isotopes of carbon and nitrogen, and those are, I'll focus on nitrogen for now. What this, if we measure this in a tissue of an organism, I get a response and a value, which then gives me an indication of the trophic position of an organism in the food chain, like how high is it or low is it positioned in the food chain. So based on this, I can map my entire food chain because I took samples from these tiny little copepods to fish, to seals, to polar bears, and then I can really match their trophic position and build this food chain based on these values. And that is so valuable for us to know, because what I'm interested in seeing is, does the pollutant in question that I'm trying to measure and quantify, does that increase per trophic position? Does it is it highest in the polar bear or not, basically? So this is why those dietary tracers are super valuable. And from carbon, for instance, we can infer where an organism has been eating in the water column. Is it ice associated? Does the carbon come from the sea ice? Is it a pelagic eater? Is it the more benthic feeder? So this also gives me an information about, okay, where in the food web does it eat? And I think this is also really valuable to know in terms of where could it then be exposed to the pollutants.
Mm-hmm. And how do you take the samples? For example, with the polar bear?
Yeah, so the polar bear, those were samples that I didn't take myself, because for this you need proper permits, you need the right training. So this was sampled by helicopter from the Norwegian Polar Institute. And then you need professionals who can tranquilize the bear, take the blood samples and the little tissue samples. So this has to be done by a very, professional people, and not something that we do on our cruises, or haven't done, at least on these ones. But what we do on the cruises is usually we have lots of nets that go in, where we then sample the zooplankton community, and we have always people who look at the biodiversity, for instance. So they map all the species that we catch, and then the rest goes in and picks whatever they need. So it's a very community effort to get the broadest picture possible, or broad and most detailed as well. And what we also did, we trawled. So then we have to, depending on the fish species that we're targeting, we had to either trawl in the water column or on the bottom. And this is always a bit, I find it challenging sometimes, because benthic trawling is very destructive, because the trawl scrapes the seafloor. So we were all trying to be very mindful of how and when we trawl and for how long, what is needed. And we all always merged the teams that use this trawl. So we worked closely together with the benthic trawl when we needed benthic fish, benthic team when we needed benthic fish. So they took samples of the community that was sitting on top of the seafloor. So that's what I also find so fascinating about doing this Arctic field work. You see how the other teams work. You see that really the entire ecosystem is, at least on our research cruises, has been looked at, and it's just so fascinating to see the others work, and then being able to share the samples that we collect is fantastic. I think that's the best use of those resources. So this is mainly what we did. We had the trawls, and we had the lots and lots of nets that we sorted through. Now,
So you said you caught a lot of different species at once. What was the broadest diversity that you had there?
To be honest, the polar night cruises, they had the most diversity. We had nets. Sometimes we went down to a couple of 1,000 meters. And then we caught this bright red shrimp, for instance. That was always a classic signature of, okay, now we're really, really deep. And then so we had one cruise where the cruise leader, she is, her main field is with copepods. So she studies copepods a lot. And she got so, so excited because it was one of the first times since she saw this amount of male Calana superboreals. So you showed a picture of the female, and apparently the males, they only exist during this time of the year. So this is what we found then, and that was, yeah, the excitement. Just that was inspiration for many days, because it was just so fun to see how excited people get about this tiny animal. So at that time, we had lots of different animals. We had amphipods and krill and these they're really cool. They're called sea angels, depending on what they look like, because when they eat, so they're little sea slugs and they are in the water column. So they kind of sit there and try to swim, and they have these little wings. But when they eat, they have this, it looks a bit like tentacles when they try to eat. It sits on top of their head. Looks very, very scary. I can share a picture with you. So these are called Cleonelle mastina, or sea angel. And when they have this, their normal shape, kind of, they're called the sea devil, because they have these two little horns. Yeah, so they change their shape and they are just, yeah, really, really cool. And they're very abundant. So we've had them in almost every net. So you see all sorts of, and that's also what I like about studying the entire food web, in a way, food chain, because when we think about the Arctic, most people associate the polar bear with the Arctic environment, and also with climate change, because we know sea ice is decreasing, the polar bear's habitat is vanishing, and the poor polar bear will starve. And while this is absolutely true, there are also so many other, maybe less iconic animals that experience these changes and these threats from climate change, from the chemical pollution, not so much in this particular area. But so I think it's so fun to, when I started studying this, to see just the biodiversity in the Arctic. It's not just the polar bear, it's not just maybe the majestic whales, but there's so much more life. Also the seabirds. It's just, yeah, there's so much going on. So yeah, that's been really fun.
Sounds amazing. How do pollutants interact with ecological stresses like food availability or prey dynamics?
Yeah, so this is also something that's quite essential, and something I maybe touched upon now already, because when so when we think about the seasonality again, and the absence and presence all of a sudden during springtime of the pollutants, we see that there's a multitude of ways that everything can interact. So one thing is that what I mentioned before is this remobilization of pollutants back into the bloodstream, then causing negative health effect potentially. But another thing is also these predator-prey dynamics, which probably change. And we see several examples where, for instance, then the seabirds, who have been specialized on this copepod that I talked about lots now, so they've been specialized on catching these. They're these tiny little birds. They're really cute, to be honest, called little auk, and they have been feeding on the Calana superboreals for a long time. And nowadays we see that they switch their diet, and these Calana superboreals, they're quite big compared to other similar species or other, yeah. So they're quite easy to find, and because they are so big, they're quite a rich meal in and of themselves already. So now that bird eats copepods that are smaller than this Calanus superboreus copepod, probably also less lipid rich, and it has to eat more. So those two factors mean that the bird has to spend more energy to get the same amount of food that it got before, or the same level of energy that it got from the Calanus superboreus. So that, of course, then has impacts on his body condition, maybe breeding success later on. So it's this cascading effect to what happens now that when we have these changing in prey and predator dynamics, will then the reduced body condition lead to higher pollutant concentrations, more toxic effects in the body, and also when the predator switches to a different prey now, maybe a more southern prey, how will that prey have maybe other pollutants that it brings with it because it comes from more southern regions, is maybe more migratory. So this is also something that I look at into the current study that I'm working on, where we integrated this subarctic fish in a very simple food chain consisting of polar cod, which is this endemic Arctic fish species, and ringed seal, which is a predator of polar cod. It preferably preys on polar cod in Svalbard, and then polar bear. And then we were thinking, we've seen in other places that the ringed seal eats this subarctic fish called capelin. And then will this have impact on its on its pollutant load in a way? And this has been interesting to see or study because I analyzed chlorinated paraffins in these samples. And chlorinated paraffins are a type of pollutant group that have been partly regulated under the Stockholm Convention. And the Stockholm Convention kind of was put in place to ban certain pollutants. But they only regulate specific parts, not the entire group of pollutants, but always a specific thing for which we have enough data. So part of these chlorinated paraffins that I'm studying right now, they are regulated. Another large part of those is not. And we see that this unregulated part is in a higher proportion in the subarctic species compared to the Arctic ones. The Arctic ones have the more regulated compounds, and the subarctic species, the newer one, the unregulated one. And that's been interesting to see because when we think about, okay, will the seal maybe in the future eat the capelin, will it then have different pollutants that it otherwise wouldn't have been introduced to? So yeah, these are all new ways where also climate change plays a role in reshaping the pathways of contaminants, because sometimes it's not just the wind or the ocean currents or rivers or glacial melt nowadays. It's also the animals themselves who move and are little like pollutant taxis almost, who go around. So yeah, I hope that answered the question.
Yeah, very well. And you just talked a little bit about polar cod. You also co-authored work on polar cod. What were the key findings regarding the synergistic efforts of food oil exposure and warming?
Yeah, so when we looked at polar cod, as I mentioned, polar cod it's a beautiful fish, really. Yeah, if you Google it and find videos or so of it, it has these beautiful like pectoral fins. It's just really, really pretty fish. It's just about this size. I think the biggest fish we measured was like 28 centimeters or so. It's not super big. So it's not an Atlantic cod, not comparable to this at all. But it plays this really big role in the Arctic ecosystem because it connects these lower trophic levels with the really high ones, with the fish, with the mammals. It's very energy dense, very lipid rich. So what also made the polar cod very interesting to study is its early life stages, so the eggs and the larvae. The eggs are buoyant, so they stick kind of underneath the sea ice. Once they're spawned, they develop underneath the sea ice. If an oil spill was to occur in that area, the oil would also sit on top of the water, close to the ice and maybe be stuck in the ice and be stuck in those brine channels. But what we see is when the embryos are exposed to the oil, at that stage in its life, it lacks any detoxification method. And it's so vulnerable because this is where everything happens. All the organs are being developed. Everything is being formed. And if this is interrupted by a pollutant like crude oil, and crude oil, I'm saying a pollutant. Crude oil is a mixture of all sorts of chemicals. So it's not a single component. But if it's then exposed to this, we see that the results are malformation of the body. So we have spinal curvatures, for instance, in those fish. They can have jaw deformities. I've sent you a picture which you could use for this as well. And then they could have the eye deformities, the heart beat, we know, is different. So the heart is impacted. For instance, in my master thesis, I looked at the behavior of the fish, where we also saw that there was a change. And what all of this tells us is just the crude oil had negative effects already on the fish survival and growth. And what we also saw when we added temperature, that this effect was just amplified. So we raised the fish at higher temperatures, around 3 degrees, which is kind of at their maximum comfort zone or outside the maximum comfort zone. But we know with ongoing climate change, they will experience warmer temperatures. So that was kind of the scenario. And then we see that the effects that we saw from the crude oil just were amplified by the temperature. We also saw that they hatched earlier, because what temperature does during the development is it makes everything speed up a bit. Everything goes way faster when the temperatures are higher. So they develop faster and they hatch earlier. And that also raises questions of, will they encounter enough food when they hatch at an earlier time? Is the spring bloom in place already? So it's a list of questions that appears when you study this kind of stuff. But it was great because the limit of lab studies is that it's really difficult to have multiple stressors, because it gets a bit much at some point. But there's a tendency for more studies to do this now. But what we did at the time, we had the crude oil. And I think what we used really, really low levels, and we saw really big effects already. I think it was compared to 4 droplets of oil in a big Olympic-sized swimming pool, which is, when you think about it, it's so little. And we saw these effects. We called them little Darth Vaders because they had this mouth malformation, so they couldn't eat, and died rather quickly after their yolk sac had been absorbed. So, with this study, the main effects was that we saw the synergistic effect. And synergistic effects means if 1 plus 1 doesn't equal 2 but equals 3, the effects that we see are amplified by each other. So this is very much what we saw. So it's just reduced survival, and size, I think, was also a lot smaller.
Quite sad. Not good
Yeah,
outcome.
and then exactly. So it's but that's also what I find challenging when it comes to ecotoxicology. It always sounds really sad, but it's not the message that I kind of want to spread. It's more like the awareness that I wish to raise with this, because there are ways to combat this and to be more mindful of choices that we make in our daily life.
Yeah.
So, yeah.
And what role do long-term monitoring and pan-Arctic data collection play in understanding contaminant drones?
So what we already see is, I think this is a question that you might also ask, is what we see is that I mentioned these legacy contaminants a lot now. So when I refer to legacy pollutants or legacy contaminants, is that they have been regulated for a longer time and banned. And when we do this long-term monitoring studies or pan-Arctic monitoring studies, we really see the effects of the regulation. So we see a decrease in the contaminant load in the animals, in the atmosphere, in the snow, the water, whatever is being sampled. And this is fantastic because it really shows, okay, once management is working, is in place, we have rules and things that have been banned and regulated, there is a positive impact. Although it takes a long time because most of these chemicals are very persistent. So even if we regulated it 20, 30 years ago, I still measure it today.
Okay.
Yeah. Yeah, exactly. So, but that just shows it's so important to have these in place and start regulating when we can, basically. Um, yeah.
In what ways is climate change expected to alter the balance between pollutant input, sequestration, degradation, and pollutant transfer?
So when I think I touched upon some of those things already, because what happens during climate change, of course, things get warmer. Um, so what we see already now is that we have, for instance, on Svalbard, we have increased glacial melt, and within the glacier, things have been trapped for a long time that are now being released into the ocean. So that changes those pollutant dynamics to have been probably kind of frozen in ice and in time, which are now being released again, which are called like secondary release. But we also see the same happening, for instance, for permafrost and what where now things that have been trapped in the ice and conserved there for a long time are being released now that everything melts. And another thing is now that the temperature is changing and elevating, and that also changes the physical chemical properties of the contaminants. It changes metabolism of the organisms. So there's so much that is, yeah, intertwined in a way, and there's an interplay between so many different things. So we can't really say accurately what is going to happen, but we see that there is an interactive effect of this temperature yeah, of the pollutants already. Yeah.
Yeah. From a scientific perspective, why is it critical to understand contaminant dynamics across seasons rather than relying on snapshots?
So what I think is that when we do the monitoring, for instance, most of the monitoring happens, or I think all of it happens in the high Arctic during the summer and spring months, which makes sense because of pure accessibility reasons, because you need to be able to go to that place, be able to do your measurements, see. It's just so much more effort to do your research into the polar night, and stuff will just be less accessible because there will be more sea ice, more everything, more cold. Um, so what I think is important, because right now we just don't know how pollutants vary across the year. We just don't know because there's no data from the polar night. So I think this is one of the gaps that I try to fill, the knowledge gaps with my research, because then we can at least make an informed decision. When is the best time to sample in the year? When when is it maybe most critical to sample in the year? And which moments could we leave out, or do we maybe need 2 sampling points per year instead of just 1? Or is yeah. So I think this is why it really matters to understand the seasonal picture and not just having a snapshot, because it varies so much, that entire system throughout the year. And depending on what animal you also include, it will change. So yeah.
I can imagine. And how do you approach communicating your research to policymakers, the scientific community, and the public?
I have always thought that, children are kind of the future. And to me, I always thought it's so important to make people care about the nature, our environment, I don't know, our home, basically. And I thought, it's probably best to start with the kids because they're the future. So I focus a lot of my outreach work on going to classrooms, trying to connect with um, classrooms, also other places in the world, using Skype a Scientist, which is a really cool platform for researchers to be able to, get in touch with other communities. So I did lots of that stuff. I also had before one of those research cruises, I had a class in Germany sending me questions that I then tried to answer while I was on the ship. So I took some pictures for them, and then I went back and then went through those questions with them. That was really cool, and I think they really enjoyed that, and I did as well, to be honest. At some point, I realized also that, now also I have kids, my own kids, and I see, yes, it's so important to get them connected to nature. But I also see that the parents have to be on board and have to kind of live that for them in a way. So I think I changed my focus a bit now. So I did more public talks where kind of science is a cool thing, for instance, for researchers, I think. You meet at a bar, you listen to, I think it was 5 minutes or so presentations, popular science talks, and have a beer while you do that and kind of chat with everyone. So it's a really low key, nice environment to do this. And so this is, I think, how I engage with the public. We also organized stands at those science fairs to kind of show what we're up to. Organize some, yeah, we provided some teaching material. We have schools visiting us on campus. We do outreach also amongst us at within our groups, our research groups. Also, while I was on board, we had an open lab day. So on the research vessel, all the scientists kind of opened their labs and invited the crew and the other scientists in, because when you're in this environment, you're so focused on your own work. You're focused on your own work, when to eat and when to sleep. Those three things, and there's not much more to it. But then we realized, we kind of want to know what everybody else is up to. So we tried to like free up our schedules. I think it was for an hour or so. Really cleared everything, and we're like, okay, we want to know what's going on. And I think this is such a great bridge, because you see how the others work, you're inspired maybe, and then it's just great to form new relationships and maybe also new projects. So yeah, so we visited each other's labs and just showed our research basically on board. And I think that's also important. And then I was part of this UN Ocean Decade National Committee, which I think we were 10-ish people from all sorts of different sectors in Norway, who kind of just spread awareness about the UN Ocean Decade that we're in right now. I mean, we're more than halfway now. And that has also been a great experience, because it's very close to policymakers, and it's also close to businesses and just people who think very differently from me. So I think that's been very enriching in a way.
Mhm. Sounds great what you have done so far, and I'm looking forward to see what you're doing next with your public outreach and communication.
Yeah,
And
thanks.
for the last question, looking ahead, what questions do you believe are most urgent for the next decade of research on pollution, climate change, and Arctic ecosystems?
Oh, it's a really, really good and broad question, I have to say. What I think will matter most, I think it's hard to pinpoint, to be honest, but I think one major thing is what we need to fix in a way is that we're able to, once we regulate something, and I mentioned briefly before that we only regulate a single chemical, but then the industry goes ahead and just changes a position. So the core kind of stays the same, but it's unregulated. They can still use it. So we have to go again and do what we did before, trying to prove, what you're doing is harmful and persistent, and please find other options. So what I think needs to happen is that we find, and I think it is happening already when we look at forever chemicals. This is the first group that is going to be regulated as a group, because it is so many different individual chemicals. And when you ban every single one, it just takes so much effort from the ecotoxicologists, chemical analysts sides. So I think this has to happen, that we finally find a good way how to regulate groups of chemicals to be more effective with the banning. When I look at climate change and Arctic ecosystems, I think it's really difficult to predict because we already see so many things happening that we thought are going to happen a bit later. When it comes to the sea ice loss, everything seems going very fast, or faster than we want it to go. So I think here we just need to be very mindful and a bit adaptive about also how is AI going to impact our research? How can we use it also to find quicker solutions maybe? And I think what I saw in The Guardian recently, they published an article saying, climate change is almost, or chemical pollution is equal to climate change when it comes to the severity. And I thought it was really interesting and fantastic, to be honest, to have climate change put on the map together with chemical pollution. And what I think we also need to include in this is the biodiversity loss that we experience, and how that is impacted by those two other stressors. And I think we need to find out we need to find out more about this. But we already have a good start, I think, as well.
Thank you so much, Julia, for having been on the podcast. It was extremely interesting to learn all about your research, and I hope to have you on the podcast soon with your new research.